Hello, everyone. Today, it's my pleasure to introduce an article to you today, titled Wuda Granule Improves the Gastrointestinal Motility via Gut-Brain Axis in Beagle Dogs. Gastrointestinal motility disorder, GMD, is a common postoperative complication of abdominal surgery, manifesting as abdominal distension, pain, nausea, etc., which seriously affects postoperative recovery. However, effective clinical interventions are still lacking. Wuda Granule, WD, a traditional Chinese medicine preparation composed of 5 herbs, Panax ginseng, Erica catechu L, etc., has been proven to promote postoperative gastrointestinal motility recovery, but its specific mechanism remains unclear. The gut-brain axis, a bidirectional regulatory network between the central nervous system, CS, and the gastrointestinal tract, plays a key role in regulating gastrointestinal motility. This study focuses on exploring whether WDG exerts progastrointestinal motility effects by acting directly on the CNS. 1. Central Wuda granule drives whole gut motility. 2. The prokinetic effect of Wuda granule is vagal cholinergic dependent. 3. Wuda granule ameliorates the structural integrity of GI cells. 1. Lateral ventricular injection of WDG in awake, fasting beagles. A dose-response study confirmed 5 mg as WDG's optimal central dose. When injected during MMC phase I in awake, fasting beagles, WDG significantly elevated contractile motility indices of the body, antrum, duodenum, jejunum, and colon, all p less than 0.05, with a more pronounced effect, 66.94% to 132.06% higher, than in anesthetized dogs. 2. Lateral ventricular injection of WDG in anesthetized beagles. Under propofol anesthesia, beagles lost periodic MMC motility, only MMC phase I-like rest. Post 5 mg WDG injection, the entire gut rapidly showed MMC phase III-like high-amplitude frequency contractions, lasting 5 to 10 minutes, with a statistically significant increase in contractile motility index versus control, p less than 0.001. 3. WDG effect post-bilateral cervical vagotomy. Bilateral vagotomy prolonged beagles' gastrointestinal MMC cycle, especially phase I, and weakened gastric body/antrum MMC phase III contractions. Lateral ventricular WDG then failed to enhance gastric body/antrum but slightly improved duodenum, jejunum, and colon motility, indicating WDG's gastric effect depends on the vagus nerve, while lower gut effects use non-vagal pathways. 4. Atropine blockade of WDG's gastrointestinal effect. After intravenous atropine, 50 μg/kg bolus plus 50 μg/kgh, infusion, fully inhibited gut contractions, lateral ventricular WDG no longer enhanced motility, confirming WDG's synkinetic effect relies on cholinergic pathways. 5. WDG's lack of intestinal structural alterations. TEM showed WDG-treated beagles had intact ileal ultrastructure, clear ICC membranes, normal mitochondria, mild rough endoplasmic reticulum dilation with ribosomes, versus controls, with no cell damage or organelle abnormality, proving WDG's safety for intestinal structure. The central administration of WDG can enhance contractile motility from the gastric body to the colon, potentially via the vagal cholinergic downstream pathway. Importantly, transmission electron microscopy analysis revealed that WDG activates central vagal pathways without inducing intestinal structural alterations, which further supports the hypothesis that its mechanism of action is primarily through the activation of the vagal cholinergic pathway. However, further experiments are warranted to determine the main active ingredient responsible for the central effect of WDG, as it is governed by the central nucleus and its impact on gastrointestinal motility.